we are looking at paper 6 version 2, so that's why it's called 62, and it's from the March 2018 exam series. So paper 6 is your alternative to practical, and so we're building all of those little bits and pieces of skills that we've learned all together into one paper. So if we look at the instructions, it is our standard instructions. Center name, candidate name, and number. Okay, you're writing in dark blue or black, so making sure that that is dark blue, not those pale blue pens, because they don't always scan very nicely. Okay, you can use an HB pencil, again, nice soft pencil for any diagrams or graphs. Do not use staples, paper clips, glue or correction fluid, no writing in any barcodes. And you must answer all the questions. You can use an electronic calculator. You may lose marks if you don't show your working or if you don't use appropriate units. Okay, so it's biology, but working and units are still really important. Okay, at the end of the examination, fasten all your work securely together, and number of marks is given in brackets at the end of each question or part question. So the first thing you would do when you open your exam paper is you just have a little quick flick through and see what it is you're dealing with. So if we go through quickly, we see we've got something going on with plants here. So this should look familiar as celery. If you don't eat it, at least you've seen it in your textbook. So we're doing something, and now this is making sense. Xylem tissue dye, okay, yeah, we've heard of this before. Got some calculations. There's our errors coming through. If we carry on, now we've got to plan an investigation. So there's that skill that they're testing. Traditional 6 marks. So there's 19 marks for that first half of the paper, the first question 1. Question 2, now we're looking at something under a microscope, and they tell us it's a bronchus. So funnily enough, here's our drawing question. And then we've got a magnification question. So the papers are very repetitive, which is what's wonderful. So if you do a lot of practice, then you've got a good idea of what's coming. Okay, then we have an investigation on breathing rate. We have a table to work with, an average to calculate, a graph to draw. There it is. So we know we're going to have specific things that we're dealing with when it comes to drawing this graph. We have a description question. We have a suggestion for safety. And we have more talk about variables, dependent, independent, constant, controlled. All of those sorts of things, and this question 2 is worth 21 marks. So we should be spending about half our time on question 1 and half our time on question 2, because the marks are split evenly. So if we go right back to the very beginning and remind ourselves, this is a 1-hour paper. So in theory, 30 minutes for question 1, 30 minutes for question 2. But it would be nice if we could do them a little bit shorter than that, so we have time also to read through and check our work. Okay, but we don't want to have at any point in time where we're just sitting twiddling our thumbs. We should be busy for the full hour of the exam. So what we're going to do in this lesson is we're going to work through the questions. So we may well finish the full question 1. It depends on how much extra talking I do. Okay, then we will continue with the paper in the next lesson or 2 lessons, depending on how far we get. So now we're ready. We know that question 1 is about plant material. There's celery, there's dye, there's things going on that are familiar, but we really need to take the time to read the question carefully and thoroughly and precisely so that we know exactly what it is we're dealing with, because maybe they've tweaked it slightly from things we've studied before, and we want to know exactly what's going on in this experiment so that we can answer the questions properly in relationship to this experiment. Okay, so a student investigated the effect of temperature on water uptake in celery stalks. So the effect of temperature on water uptake. Okay, so already we're thinking independent, dependent, which is which. So remember, the effect of the independent. So temperature is our independent variable on water uptake. That is then our dependent variable. Water is transported in the xylem tissue in plant stems. Yep, we're happy with that. Okay, step 1. One celery stalk was placed into a beaker containing warm red stain. Another celery stalk was placed into a beaker containing cool red stain.
How much red stain? It didn't tell us. Okay, it didn't tell us where the celery stalks came from. Are they off the same plant? Okay, so we'll keep that in mind. Okay, shown in figure 1.1. Here is our xylem tissue in the little circles. We have our celery stalk, the beaker, and the red stain. So this is either warm or cold. But again, we've got this issue of, remember when we work with temperature, we want to make sure the temperature is kept constant. And there is nothing here that is maintaining that temperature. Okay, so that's another thing that we're keeping in mind here. We're not entirely happy with just having different temperatures in beakers. Okay, step 2. Both celery stalks were left in the red stain for 10 minutes. That we like. Both of them. They both had the same thing done to them, and it's an exact 10 minutes. Okay. After 10 minutes, the celery stalks were removed from the red stain. Perfect. Both of them again. So they were both in for exactly 10 minutes. Step 4. A 5mm section was cut from the end of a celery stalk which had been in the warm red stain, as shown in figure 1.2. Okay, so we're starting from the bottom that was sitting in the stain, so this bottom here, okay, and we're cutting just 5mm. So that length there is going to be 5mm. Okay. The section was inspected for the presence of the red stain in the xylem tissue in the celery stalk as shown in figure 1.3. So there's the xylem tissue. So this is now the cross section of the celery stalk. And we're saying, do we have red visible in this xylem tissue? Yes or no? Okay, so we're inspecting. If the red stain is visible in that cut section, another 5mm section was cut, and the process repeated until no red stain visible in the cut section. This allowed the student to estimate how far the red stain had moved up the celery stalk. So they're just going to cut and then inspect, cut and inspect, cut and inspect, okay, until there is no red stain. Steps 4 to 6 were repeated for the celery stalk which had been in the cool red stain. Okay, so this is a little bit dodgy now, because how long did they spend with the one that was in the warm red stain? So this cool red stain is just lying there waiting. Okay, so that's not all too lacquer, because the timing now is different, because we've been working just with the warm and not with the cool. So we'll keep that in mind. Okie dokie, so first question, A part 1. State one safety precaution that should be taken while carrying out this method. Well, we've got 2 things that are particularly problematic in this one. One is the and the other is the cutting. Okay, so whenever you are working with any kind of stain, you must wear gloves. Okay, so wear gloves. So the stain doesn't come into contact with your skin. Okay, if you want to talk about the blade, then you could talk about how you should cut on a surface. Okay, you don't want to just be randomly cutting celery. You must have a white tile, and you must put the celery on the tile, and you must cut down. So you're keeping your fingers away from the blade. There is another safety precaution. Okay, so you've got a few safety precautions to think through. The question says state 1, so you only state 1. You're not going to get extra marks for writing extra precautions. In fact, you are then not following instructions, which is a bad thing. Okay, so pick the one you want and you write it in. Okay, the suggestions come through of wearing a lab coat. That is pretty much taken as a standard. Okay, so whenever we're in the lab doing an experiment, we'd be wearing a lab coat. So what we're looking for is, in this kind of a question, it's the particular safety precautions for this method. Is what would be special in this in particular method. Okay, the suggestion also came through of protective eyewear, so wearing goggles or glasses. This one, we're not working so much with things that are splashing. Okay, we're not working with boiling solutions, like if we're working with Benedict's.
So here it's really about the risks. In this one, you want to identify the risks of the question, and the risks are the stain and your hands, because your hands are the ones close to the stain, and then also, of course, dealing with a sharp blade. Okay, so A part 2. The students' results are shown in figure 1.4. Number of 5mm sections cut from the celery stalk in warm red stain, 11. Number of 5mm sections cut from the celery stalk in cool red stain, 5. Okay, and they put it like a little excerpt from a notebook. Okay, it isn't, if you're doing the experiment, then you're keeping rough notes on the side, and then you're going to turn that into a table. Because sometimes you need to sort of think through what's my results, then how am I going to build my table? So that's the idea here between this notebook excerpt. So we are then asked to prepare a table and record your results in your table in the space provided. The table should include the temperature, cool or warm, we don't know the actual degrees temperature of the red stain. The number of sections that were stained in 10 minutes, the total distance moved by the red stain in 10 minutes. So there's 3 things we must include. That means we're going to need 3 columns. Okay, remember when we're doing this, we're drawing our table in pencil with a ruler. Okay, I don't have a ruler here, so I'm going to get away with not using a ruler, but you guys don't have any excuse in your exam. You must have your ruler. Okay, so we wanted 3 columns. Okay, remember your independent variable always goes in the far left, and then we have measurements of our dependent variable towards the right. Okay, we have quite long headings for these, so we'll make a nice big space. And then we've got 2 different options. So our table is built. Now we can go back to our pen to fill in our table. So independent temperature. So we have temperature of the red stain. Okay, and it can either be cool or warm. Okay, so if we look at our results, we have warm on top of cool, and we did the warm first, so we should have warm first in our table. So warm and cool. Next one is number of sections that were stained, and that is then what we're going to have to put. Number of sections stained in 10 minutes. Okay, so for warm, there it is, it's 11. For cool, here it is, 5. Okay, so then what's our last column? Is that total distance moved in 10 minutes. So we need distance moved by red stain in 10 minutes, but this is now a distance, so we need a unit. So we put our slash, and what are we going to be having our units? Remember, we cut 5mm slices, so our units for this is going to be millimeters. Okay, so now we have to think, right, so there's 11 sections. Each section was 5mm wide, so the distance then is going to be 11 times 5, which is 55. And we don't put any units here because the units only go in the heading of the column. So then the same thing for our cool. We had 5 sections, each was 5mm, so that's a total of 5 times 5, giving us 25mm again. Headings are in, units are in there, heading only. So now we have our table, our results table, all filled in and ready to go. So what is the next question? Use the data that we've got on our table to calculate the rate of movement of the red stain in the celery stalk at each temperature. So one for cool and one for warm. We have space for our working, and they give us a lovely clue here in the answer space. They say warm blank millimeters per minute, cool blank millimeters per minute. Now millimeters per minute means we're going to have millimeters on top of our fraction and then minutes underneath. So for warm, we have 55 millimeters in 10 minutes, which is 5.5 millimeters per minute. Okay. For cool, we have 25 millimeters that we've moved in 10 minutes. So we have 2.5. So cool would be 2.5 millimeters per minute. So we can see that our warm has moved further, but because it's also only in 10 minutes, we kept them both in that same just for 10 minutes, we have a faster rate of movement in the warm temperature. Okay, so what comes next? Funnily enough, once you got your results, now you need a conclusion. So as we've just discussed, the conclusion is going to relate to the temperature because that's what we're testing. We're testing the effect of temperature. Okay, so we go right back to the beginning. We wanted the effect of temperature on water uptake. So something about how temperature affects the rate of uptake, well, that would then be the stain moves faster, so water uptake is faster at higher temperatures. Okay, so there's lots of different ways that you could phrase this, but the idea is that when the stain was warm, it moved faster. It moved further up in the same amount of time. Okay, so now talking about variables. Two variables which were kept constant in this experiment, just two of them. Now remember, we've thought about it now several times. Both of them were kept in that stain for 10 minutes. So time in the stain. Okay, that was 10 minutes. Another thing that was kept constant, well, the other thing that we did that was definitely the same for the warm and the cold was we cut them at 5 millimeters. Each of them was cut at 5 millimeters, 5 millimeters, 5 millimeters. Okay, so the width of the cut pieces was kept constant. Also the type of plant. We use celery in both beakers, okay? We had a suggestion come through about the concentration of the red stain. Well, we weren't actually given information. We weren't told that the same red stain was put into the two beakers. So it's a little bit of an assumption that they've used the same concentration. We would expect them to, but it's not actually stated in the method for this experiment. Okay, so another thing that we sort of assuming, but they didn't state, is things like we've taken the celery off the same plant, that there's the same amount of stain in both beakers. Okay, so we assume that they've put 100 mils in each beaker and it's the same stain, but they didn't actually tell us. So when it comes to stating our variables that are kept constant, we want to rather stick to the ones that are specifically kept constant, that we're told about in that method. Okay, so one way of improving the method used in this investigation would be to repeat it a number of times. Identify two other sources of error in this investigation, and for each error suggest an improvement to minimize its effects. So there were two things that we were really unhappy with when it came to this experiment as we were reading through. The first one was we said that if we're working with temperatures, we want more than just a beaker. Okay, so the first error would be then that the stain can change temperature through
the course of the 10 minutes. And why is that a problem? It's because warm is not necessarily warm anymore. Warm could be now at the end of 10 minutes very much more similar to the cold. So how do we keep temperature constant? We're going to use a water bath. At an appropriate temperature. So you would have one water bath that is at a warm temperature and one water bath that's at a cool temperature. Another error. Okay, so we have sort of issues about the fact of volume of stain and things, but it's not really an error because we would have expected them to do the right thing. The thing in the method that is a problem, of course, is remember we said we're going to cut this warm wine and we're going to cut and look and cut and look and cut and look. And I mean, at the end of the day, we found out they cut it and look it 11 times. So cut and look, cut and look. Now, 11 times is actually a substantial amount of time. Meanwhile, that one from the cool one is still potentially moving because what's saying that the xylem has stopped? It didn't freeze anything in time. That water can still be moving up and therefore that stain can still be moving up the xylem in the cooler one. So the stain, error is that stain in the cool, stain in celery from cool beaker can still move while cutting the warm beaker celery. Okay, so concentration of the red stain would potentially be another error, but not really because we would expect them to be the same. Okay, so we would expect them to make one batch of stain and then use it between the two. So what we're looking for is specific errors in the method. So what is definitely a problem? And there's definitely a problem here with not doing it at the same time. What we should have done is cut at 0.5, look at them both. Cut at 0.5, look at them both. Sorry, 0.5 centimeters, 5 millimeters. Okay, so the improvement to actually get rid of this error completely is not you should have told us that we use the same concentration stain. It's the idea that, you know, we should have cut them both together and looked, and then they're both sitting for exactly the same amount of time. Okay, so the improvement here is cut both stalks and look for stain at the same time. Alternatively, you could do warm 10 minutes, cut and look. Then cool 10 minutes, cut and look. You could do them completely independently. But if they're going to be sitting in simultaneously, they must be then cut simultaneously. So that we're doing the same thing. Okay, so we don't have much more time left, and the next one is to actually build an experiment. We want to have time to properly think through that one. So we're going to save that one for next time. So just a recap now of what we've covered in this session. We've gone through our experiment, reading it and making sure that we really understand what is going on. Okay? At the same time, we are looking for those gaping holes, the glaring errors, and the things that we really like. Because the gaping holes, the glaring errors, those are the things that we're keeping in mind for errors. And the things that we really like, those are the things we're keeping in mind for control. So for temperature, it's all about insulation. For timing, it's all about consistency. For cutting,
all about measuring the same thing and then doing things simultaneously so you keep that same time going. Okay, then we looked at results table. So it's a reminder, units only in the headings. Proper headings, exactly as they tell us there in the instructions. Always independent variable on the left-hand side, then dependent measurements on the right-hand side. Calculating a rate, it's movement over time, and we can use the units in the answer to help guide us on that calculation. Fantastic. Once we've got results, we can move to conclusions, but we want to bear in mind what was the original question of the experiment before we then phrase our conclusion, so that we get a conclusion that ties in with that original aim of the experiment. Okay, then they ask us about variables, and because we've thought through the experiment properly, we've understood what's going on. We can set our variables, and we can do the same thing then with thinking about our area. We're thinking about our errors. What are the things in this method that are definitely a problem? Not the could be a problem one, or we're not sure that we like your instructions here, but what are definitely the problems with this method as we give them?